Welcome to Progenesis Academy. Today we are celebrating webinar number 50. Since the last 18 months, Progenesis Academy has been focusing on educational webinars for the embryology community. We are so fortunate to have many embryologists and laboratory directors who have sacrificed their personal time to provide us with high quality educational material. For those who are not familiar with Progenesis Academy, you can find our webinars on Progenesis website, progenesis/academy, or on our YouTube channel, Progenesis Academy. If you subscribe, you will get notification when new content is available. Today's topic is FDA requirements for sperm donors, egg donors, and tissue bank. We have an amazing panel of experts, laboratory directors who are going to help us understand the FDA requirements. Introducing Marlene Engel, she is a lab director at Generation Fertility Center. Ammon Baylis, he is the lab director at Utah Fertility Center. And Carly Chapman, she is a lab director at Reinhardt Fertility Center. Marlene, the floor is yours. Thank you so much.
Thank you. You can go to the next slide. And you just keep going. So I am presenting this really from the standpoint of the laboratory. And I think every laboratory gets a little bit more involved or less involved depending on the lab. But one of the reasons we have to interact with the FDA is because of CFR 1271, Title 21, human cells, tissues, and cellular and tissue-based products. Because of this law, in subpart B, the law says all labs who handle donor tissue, in other words, anyone who is recovering, processing, storing, labeling, packaging, and distributing are required to register with the FDA. You have to do that by the end of December every year, and it's all online. There's no charge. And this tissues and that we are directly applicable to what we do and use would be donor sperm, oocytes, embryos, testicular, and ovarian tissue. So I'm the one who does this for most of my labs, but I know that's not necessarily the case for everyone. So I'm the one who goes in and registers, fills out all the paperwork, and makes our declarations for the year. Next slide, please. Nabil, could I have the next slide, please? Thank you. All right, so that's part B that defines what we have to do. Part C of the law discusses donor eligibility. We don't, in most of my labs, we don't get hugely involved in determining donor eligibility. This tends to be a doc or nursing role. But what the law says is the clinic must screen the donor, whether it's male or female. So you have to come up with a questionnaire to assess lifestyle, where they have been in regards to Zika or some of the things that we can't do an infectious disease test for. You have to assess exposure risk, and the full screen has to be done once a year, and then you can do an abbreviated one every 6 months. The clinic also has to do a history and physical for potential risk factors. This has caused us problems in some of my labs where it's primarily OB-GYNs or REIs that are running the lab or running the clinic, and they don't want to see and do an H&P on men. They feel it's outside their scope of practice. So for many of those, we have to send them off to their PCP and get them to do the exam. But for all of our female patients, our REIs will do the H&P and all the screening. And then you have to test for infectious diseases. And I think that list is going to be in Eamon's presentation. So I'm just going to go on to the next slide, please. and not specifically discuss all the diseases we have to test for. One of the things that's hard for our clinic is that the FDA requires written procedures for testing, screening, determining donor eligibility, and complying with all other requirements of the subpart C. I find this is hard for the clinic because most docs and nurses aren't used to writing clinical protocols. That's something obviously we as lab people do all the time. But what I often do is write a template of the protocols because I don't know exactly what each one of my labs does when they're doing how they're doing the testing, what their workflow is, when they're requiring screening, when they make the determination. So I come up with a template for my labs that they modify based on exactly how they do this. And then I usually go back and force them to update it every year and sign it, that sort of thing. Next slide, please. However, where we do get involved is in a review of the summary of records. So the FDA requires that at the completion of the screening and testing, a summary of records has to be completed by the physician, stating whether or not the donor is deemed eligible by the physician. So the clinic gives us a completed SOR after they finish screening and testing, and we usually require that this is done and in our hands by the day of egg retrieval. Although technically you have 30 days prior to an oocyte retrieval and 7 days on either side of sperm retrieval and cryo in order to get that infectious disease testing done. I know our clinic staff ask for infectious disease testing, but it's amazing how many of those tests or how many patients are missing a test and things just slide through the cracks. So we view ourselves as being the final stop along the way, and we get the final right of refusal, or we force them to bring the patient back and get that testing done before egg retrieval if they haven't done it. For sperm, you need to get the infectious disease testing drawn within 7 days of sperm retrieval. If it's donor sperm, sperm has to be quarantined and the male has to be retested at 6 months. We don't do any anonymous donor testing. We purchase all of our donor sperm from registered donor egg banks or donor sperm banks. Same with most of our eggs, not all of them. But we definitely are not doing the quarantining and infectious disease testing for sperm donors, anonymous donors. We will do this for donated donors, directed donors. Next slide, please. So we review the SOR prior to oocyte retrieval and prior to thawing the sperm or any other frozen tissue to make sure it's been deemed as eligible. But one of the things that's nice for us in Wisconsin is our clinic works with Eversana to help manage the testing and screening and documentation. So they have been a lot of help, and they work very closely with our third-party coordinator to make sure all of this workup is being done. Next slide, please. And they do a great job. All right, where we do get involved is at the end of labeling. So the FDA requires that all tissue be labeled, and the label should indicate the FDA status. If there's any kind of issue, the FDA law says you must prominently label an HCTP made available for use under the provisions of that paragraph. And the biohazard legend has to be on the label. And there has to be this warning, advise patients of communicable disease risks. And in the case of reactive test results, you have to say warning reactive test results for, and specifically identify what the disease agent or disease is. Obviously, all of that's not going to fit on one of our cryo labels. Next slide, please. So that was early on, that was a huge issue with how are we supposed to label these devices. And it finally became obvious from the FDA that we didn't have to fit all of that information on the device, whether it was a straw, a vial, Vitrograd, Cryolock, whatever. This can be a piece of paper that accompanies the patient chart, but it must be accessible and reviewable whenever a sample is warmed and thawed and when the FDA comes in for inspection. That's been a huge issue here at the University of Wisconsin. We work with Epic as the EMR, and our Epic people just don't want to scan in all of the paperwork that comes from the donor banks. They view it as shipping documentation, and it's taken me a year to finally make progress on convincing the Epic people that this is not a shipping document. This is information that the FDA requires us to have and wants to see. So we have to have this donor bank's summary of records, and they usually send us all the infectious disease testing. So as far as I'm concerned, and the genetic testing. So as far as I'm concerned, we should have that available also, but that's not required by law. Next slide, please. For sexually intimate partners, there is no requirement for SIPs to do any of this. And nor do directed donors have to do this. So for example, gestational carriers. I know not everyone does gestational carriers. We do a lot of them, my lab in California. However, we usually still have SIPs, directed donors, and the GC themselves do the ID testing, infectious disease testing. Because if we ever send embryos or oocytes or sperm to long-term storage, patients will pay less money for the storage if they've had testing done, the infectious disease testing, so they can go to ReproTech or California Cryobank for long-term storage and can have documentation that they're ID negative. So we do ask our sexually intimate partners and our donors and GCs to do their directed donors and GCs to do the infectious disease testing. We don't make them go through Memorial, although it's become obvious that if they do the testing at the University of Wisconsin, they're going to pay more than they would if they did it through Memorial. So a lot of patients just elect to go through Memorial, but it has to be an FDA registered and approved lab that does the testing for the donors. Next slide, please. So one of the things we were asked to discuss is what happens at FDA inspections. Usually in most of my clinics, this is run by the clinic staff. The first thing we tell everybody is, you put them in a room where they don't have unlimited access to all the charts, and you get them the chart they need. So the inspector is given a list of all the oocyte, sperm, or embryo donors that were used since the last inspection. We don't provide unlimited access to all of the charts in our history, just the last 2 years. The inspector will pick some if it's a small lab, or ask for all. Well, ask for all if it's a small lab, or pick some if it's a bigger, busier lab of the charts they want to review. They usually don't pick a lot of the sperm donor, because if they know we're not doing our own in-house sperm donor, anonymous sperm donors, and we're only getting them from banks, then they usually ask to see a list of the banks we use, but won't ask to see everything that every sperm donor. But I have seen FDA inspectors come in and go through every single one of our charts, because unlike a CAP inspection, for example, this isn't just one day. An FDA inspection can last 2 or 3. Next slide, please. So I just wanted to take a minute to talk about a couple of the issues we've had and how we dealt with them. We had one inspector who came into a new lab and wanted to inspect the lab for Part D of 21 CFR 1271. And this is the good tissue practices portion of it. And, as embryology labs, all of us are doing the things that are listed in good tissue practices. We're doing all the equipment verification and validation. I just didn't have them written up as a specific protocol to hand to my FDA inspector, because I wasn't expecting that. So I pushed back and said that it was my understanding that we were not subject to being inspected under Part D, the good tissue practices. And the inspector argued with that. So we both went to our respective computers, and I talked to our regional FDA director, and she took it up the chain on her end. And then she finally came back and rescinded the request and said, that's correct. We are not obligated to be inspected under Part D, the good tissue practices. So the point I want to make here is that the FDA inspectors are not gods, and you can push back on what they're asking from you, but there are some places where they draw a line in the sand. For example, in one of my labs, the screening paperwork was 5 pages long, and the 5th page had gotten lost someplace, and that happened to be the Zika screening portion of it. So all the subsequent forms that were copied were only 4 pages long. And the docs spent a lot of time saying, this was just simply an oversight. We had it on the original. I can pull it off for you. He printed it. He said, I don't know where the 5th page went, but she said, oh, well, that's your responsibility. Too bad. She did not rescind that request. Next slide, please. So in another instance, an inspector wanted to go into my embryology lab and wanted to see how embryo and oocyte devices were stored, labeled, and quarantined. I did not take her into the lab. We brought all the protocols, copies of all the labels, copies of all the procedures and the notebooks out to her, and we rolled a tank out into the hallway for her to look in, but I also refused to remove samples from the tanks. And she ended up being, that inspector ended up being fine with that. But I think you don't have to give them free access to everything in your lab. I think that's always something we can fall back on. I think that is my last slide. Oh, sorry, one more, bottom line. I do think inspections have gotten easier over the years because most inspectors now usually know what to look for and what to ask about, and most of us know what they want, so we've got it ready. And what we have found in several of the smaller places is our inspector is usually the same person from inspection to inspection, and so that helps. They will often just come in and check the things that we hadn't done perfectly the previous time. So I think it's generally, these inspections are generally much less confrontational these days than the first few I went through, and probably on both sides. I think the inspectors didn't really know what to do when they first started having to do this, and we certainly didn't know. And so I think it's gotten better on all sides. So the bottom line for me, I would say is, as the person being inspected, be polite, make sure you know the law, and be firm if they push in areas that you think they shouldn't. Now, pretty sure that's my last slide.
Thank you so much. Our next speaker, Ammon Bayliss. He's the lab director at Utah Fertility Center. Ammon, thank you so much.
Yeah, thank you. You can go ahead and transition to the next slide. So I'm honored to be part of this webinar for the Progenesis Academy. A little bit kind of maybe feeling a little inadequate in the FDA and navigating the requirements. It's a very challenging area. I find I don't have near the experience that Marlene does, and Carly as well. They've been doing it longer than I have. So I accepted the invitation knowing that it would benefit me. So I'm being selfish. So I had to kind of look into some things and learn some things that I need to be better at. So I think it's something we can all work on. But go ahead and transition to the next slide, please. So I think one of the main takeaways from the FDA and their requirements is the complexity. I'm just very pointing at one small aspect of it here with egg donors that we do quite a bit of here at the Utah Fertility Center. And it's a process from recruiting the donors, screening them, first getting them in, and all angles are very complex, and it takes some time, and there's a lot of hands in the pot, and so like things can get get complicated in a hurry. So I think it's very important to be very detail oriented when you're coming to anything with the FDA and your protocols, writing down what you say you'll do and then doing what you say you did, and being very thorough in that aspect. But very complex. So next slide, please. One thing I have learned here in Utah, I'm not from Utah, but Utah by far has some of the worst drivers I've ever seen. And one thing I learned quickly was when I'm driving, you got to stay in your lane and keep your eyes up for people drifting into your lane. And I carried that over into lab work, because as an embryologist and the lab in the fertility center, we really have to stay in our lane. And not be poking our nose into the third party team or the physicians, trying to make too much, or making determinations on donor eligibility that we have no business doing. So Marlena brought this up as well, but we basically follow the directive of the physicians and what they deem as eligible or not eligible based on the testing that they have ordered. So we let them do the screening, the testing, and to determine that eligibility. And we basically document and label what we need to, and then follow through. So very important to stay in our lane. So as a lab, go ahead and transition to the next one. Bill, thanks. So part of the lab's responsibility in the FDA requirements are, following the good practice, good tissue practice, like we all, I think, should be doing this, and I'm sure people are at varying degrees, but this again is a very complex requirement, and they're just a part of it. And like was mentioned earlier, nothing that we're really inspected on, but it's something that we're all doing, and it's part of the process. I think the biggest and most important part of the FDA is that kind of down on that, the second main big bullet is that no cells or tissues should be sorry, I misspoke. But nothing should be done that should spread the communicable disease. So everything we do needs to be done in an aseptic technique, process, and storing and handling and testing, so that we're not spreading our transmission of any disease. And no cells or tissue may be distributed until the physicians deem the eligibility status after the screening process. One aspect of the lab too that I think is very important is that we have a compliance officer here who has had lots of years of experience with the FDA, and she oversees all of our policies and our forms. She reports to the medical director any kind of issues that we're having with labeling or testing or screening, and she's also involved in training the staff as well in the lab, because a lot of this is kind of a different language and kind of a different thought process when it comes to determining eligibility. And she also will audit our records on occasion to see if there's things that are being missed, either by the third party team, the screening, or from the lab in the labeling, like the label that we use to identify the FDA eligibility. Are we doing a good job? Next slide, please. So this is just an example of the donor summary of records that the lab receives. So, again, we need to receive this prior to the day of the oocyte retrieval. And then this will direct us on the eligibility status of those eggs. And so just an overall example that we use then to take this donor summary record and then label the FDA labeling sheet that's attached to the cryolog, so that those samples are appropriately labeled. An example of that's on the next slide, if you'll please press forward one. So this is an example. The patient label is actually above that line. might have saved it incorrectly, but this is just an example of the FDA label that is attached to our paperwork. Whether it's sexually intimate partners, a donor embryos, or whatever the gamete source or the embryos, we attach this to all of our samples, whether it's donor or not, for the FDA labeling purposes. So it has all the required fields to indicate the eligibility status of those samples. So I think my kind of presentation was very, very short. There's a lot I think we could have gone into and talked about with the FDA. I think this could be probably 10 presentations in and of itself because it's so complex. So I really just tried to stick to the bread and butter that I'm familiar with, which is oocytes and labeling them from the donor screening standpoint. And again, thanks for letting me be involved in the bill. And I believe that's my last slide. Yeah.
Thank you so much.
Yeah.
Our next speaker is Carly Chapman. She's the lab director at Reinhardt Fertility Center. Carly, the floor is yours. Thank you so much.
Okay, so thank you, Marlene and Eamon. I am just going to recap a few kind of critical things and then do some scenarios to see how things should be labeled. Next slide, please. So 21 CFR 1271 says that if you're a donor, you have to review medical records for risk factors and evidence of RCDAD and the risk of xenotransplantation. There are additional requirements for leukocyte-rich HCTPs, that's HTLV1 and 2 and CMV, and there are additional requirements for reproductive cells, chlamydia and gonorrhea. Next slide. So the relative communicable disease agents and/or diseases are called RCDAD. Specific RCDAD requirements may change with emerging threats. For instance, there were Zika virus requirements implemented. There were West Nile viruses implemented. Next slide, please. But they also change requirements. For instance, people that have received clotting factors are no longer required to be screened, or they are required to be screened, excuse me. They're not required to be determined ineligible because they've received a clotting factor. Next slide. So clotting factors are no longer considered to be a risk for HIV, HBV, or HCV. Donors that have received clotting factors as a result of hemophilia or an acute bleeding episode, or persons that have slept with persons who have received clotting factors should no longer be asked those questions and should no longer be determined ineligible based on those questions. Next slide, please. So here, this slide just lists the agents that need to be tested and screened for. So screening is a review of medical records, your medical history questionnaire form, and testing must be FDA approved, licensed, approved, or cleared test. So your infectious disease panel isn't always a licensed, approved, or cleared FDA test, and donor testing must be done with licensed, approved, cleared FDA tests. And testing may require more than one test. For instance, HIV has an antibody test and a NAT test. So it just has to be tested for, but you have to do the test in all the ways that the FDA says you need to do it. And CMV is something that we are required to test for per the FDA rules, but it's not considered an RCDAD. Next. Okay, so there's FDA guidance and then there's ASRM guidance. So in 2021, ASRM came out with new guidance related to gamete and embryo donation. One of the things they want to start doing is use the word non-identified in lieu of anonymous. I could never remember that, so we call our donors directed and non-directed. The FDA does not require that directed sperm be frozen and quarantined, but the FDA recommends that it be frozen for a minimum of 32 days. The FDA does not require screening and testing of SIPs or the gestational host, but ASRM recommends screening and testing of SIPs if using and gestational host. Now, screening and testing with an ID panel, I want to say again, is different than donor screening and testing. Donor screening and testing must be done with FDA approved, licensed, cleared test. Next slide, please. Okay, so 2019 was the most recent year I could find. The most common reproductive tissue citations were failure to adequately screen and or test for West Nile virus. West Nile virus is one test that is seasonal. I think it's June 1st to October 31st. So maybe that's why that often gets missed. We test all our donors all year round, so we don't ever miss a West Nile. Adequately screening for Zika virus. If someone has traveled and Zika is prevalent, the FDA literally expects you to go to a CDC Zika map and look at the map for the area and determine what color the prevalence of Zika is and make a determination. The testing is performed with diagnostic test kits instead of FDA licensed, approved, or cleared test kits. The timing of sample collection, inadequate SOPs, DE not performed or not documented, and then DE not performed for directed donors. Because a donor is directed, it does not eliminate the need to screen and test that donor in exactly the same way you would screen and test an anonymous donor. Directed donors are donors, and they require the exact same screening and testing as anonymous donors. Next slide, please. Okay, so Bill and Mike are a same-sex couple, and they are the intended parents. They created embryos using an anonymous oocyte donor and Bill's semen. Bill tested reactive for hepatitis C. The resulting embryos will be transferred to a gestational carrier. Okay, we have 4 people in this scenario. Who requires donor eligibility determination? What screening and testing are required? Next slide, please. First, okay, who is a survey question? I don't know how we're going to do a survey. Who requires donor eligibility determination in this scenario? Bill, Mike, Bill's sexually intimate partner, the donor, or the gestational host? Bill, the donor, or a gestational host? Bill and the donor only, or Bill cannot be used as a semen provider because he is reactive for Hep C. So the real answer is just Bill and the donor. Remember, the FDA's guidance is to protect recipients. They do not care about the recipient's status. They are trying to protect the recipient from getting a disease from an HCTP. Next slide, please. Okay, so this scenario has an anonymous oocyte donor. So they have to have a physical exam that does not show any signs or symptoms of communicable diseases or risk factors. They need a medical history interview, and they cannot have medical history or social behaviors considered to be at risk. They have to have testing for the RCDADs listed, and they cannot be reactive. And they have to be eligible to be used. You cannot use an anonymous donor that eligibility determination is ineligible. You cannot use them. And for labeling, there's no applicable warning statement that needs to be associated with an anonymous oocyte donor because they're eligible, or you should not be using them. Next slide, please. So Bill is the semen donor. So he needs to be screened with a physical exam and a donor medical history interview within 6 months of semen recovery. RCDAD testing up to 7 days before or after the recovery of the semen. Because Bill is HCV positive and answered yes to the male-to-male sex questions, he is ineligible. There is nothing that can be done to change his status as ineligible. But he can still be used because he knows the gestational host. You cannot use a gestational host without there being some sort of relationship between you. So you are known to each other. And the SOR for Bill must state that he's ineligible. Next. So Bill's Mike, ASRM would recommend infectious disease screening, but the FDA makes no recommendations for SIPs. Mike does not need to be screened and tested per the FDA. Mike should be screened and tested per ASRM guidelines. Next slide, please. The gestational carrier. She's the recipient of the embryo created from an eligible donor, oocyte donor, and an ineligible sperm donor. FDA has no guidelines for screening and testing of recipients. The ASRM recommends a comprehensive screening for gestational hosts, which would only make sense. But in your SOPs, you need to clearly define what are you doing with your gestational host. And there's a difference, I want to just say it again, say it a million times, between an infectious disease panel and FDA cleared, approved, or licensed donor testing. Next slide, please. Okay, so in this scenario, we have an anonymous donor that requires a physical exam, medical history, testing, must be eligible, a summary of records with no special labeling. The sperm donor requires a physical exam, a donor medical history interview, RCDAD testing, must be determined ineligible. He needs an SOR, and the SOR needs to say why he's ineligible. Bob requires nothing, and the gestational host requires nothing. Next slide, please. How must frozen embryos created for Bill and Mike be labeled? Next. We need a biohazard legend that indicates the presence of a risk factor for RCDAD. So you need a biohazard label. Next. You need the statement, advise patient of communicable disease risk. Next. You need a warning statement, reactive for hepatitis C, reactive for hep B, whatever they're reactive for that is making this donor ineligible, you need to say what the warning is. Next. Okay. Bill and Mike have changed their mind, and they want to use Mike's cousin, Ann, as their oocyte donor, And they're still going to transfer the embryos into the gestational host that they had previously selected. The gestational host does not know Anne. Next slide. What type of donor is cousin Anne? Is she considered a directed donor because she is the cousin and knows Mike and Bill, or is she an anonymous donor? Next slide, please. If an oocyte donor is not known by the recipient, the gestational host, the oocyte donor must be considered anonymous. Donors can only be classified as directed if they are known by the recipient of the HCTP. But in this scenario, Anne is known by the intended parents, but not the recipient. An anonymous donor eligibility determination must be made, and it must be eligible, or Anne cannot be the donor. So the next scenario is Pam and Bill. Pam and Bill are a heterosexual couple. They have, they're using Jane, Pam's cousin, as an oocyte donor. And Jane traveled to an area where Zika is prevalent. So the donor is directed, but she has traveled to an area where Zika is present. You still need to screen and test the directed donor exactly the same way that you would screen and test an anonymous donor. However, her eligibility determination will be ineligible, and we need to warn the recipient, and we need to say what tests she was reactive for or why she is ineligible. She's ineligible because she traveled to an area with prevalent Zika. You would also need a biohazard label on that. So the difference between a directed donor and an anonymous donor is a directed donor can be used even if determined ineligible, where an anonymous donor cannot. So here's another scenario. Jane and Bob created embryos using an eligible anonymous donor and Bob's sperm. Jane has given birth to 3 children over the course of 5 years, and their family is complete. They still have embryos remaining in storage, and they wish to donate them anonymously. They don't want to know who gets them. They don't want who gets them to know. The FDA refers to this as subsequent donation. Next slide, please. So the whole point of 1271.9 does not require screening and testing of SIPs. So more than likely, these embryos, which were created with Jane and Bill or Bob or Ted's Big Adventure, weren't tested at the time they were created. So you need to consider each gamete donor individually. Eligibility determination could say not determined. This is the slide that was out of order. And you need to advise the recipient of communicable disease risk. Subsequent screening and testing, meaning at a period of time outside the window of the FDA, 30 days, 7 days, is highly recommended if you're donating embryos, but you can't force someone to do it. Next slide, please. So if no further testing occurs, you need to have a biohazard legend. You need to state that it was not tested for infectious substances, even if they had an ID panel, because it wasn't an FDA donor test and it wasn't tested in the time frame. And then warning, advise recipient of communicable disease risk. Next slide, please. Skip that slide. Okay, so Bob undergoes complete donor screening and testing, and he's negative for all risk factors. This goes back to Bob and Pam, who used an eligible donor and Bob's sperm to create embryos. Bob subsequently has complete donor screening and testing. He has a history and physical. He does the whole protocol. Does that change the labeling? Do you still have to have warning, advise patient communicable disease risk? Do you still have to have a biohazard symbol? Do you still have to say testing was not done? Next slide, please. So this is a survey question. Just think about it. How does the labeling change? It can be changed to eligible since all tests were negative. It can be changed to delete the warning statement. The biohazard sign can be removed, or there can be there's no changes on the labeling. Well, the reality is, next slide, please, there is no changes on the labeling. You can't, it's still a biohazard. It still has to say it was not evaluated for infectious substances because it wasn't done in the proper time frame. And it still has to advise recipient of communicable disease risk. And I think that's my last slide. Questions?
Thank you so much. Thanks, Carly. It was a great in details presentation. Thank you so much. Okay, let me get the questions that we had on from the audience. I think we have 3 questions so far. So let me see if I can pull these questions. As is here. Okay, so.
Okay, I'm sorry. I've been like typing answers to questions.
Yeah, no,
I don't know if I answered all the questions. There was one question about international donors, but I don't know what I did to it.
Would you like to answer this?
I will answer it. It's kind of a conundrum because international facilities don't necessarily use FDA licensed, screened, or cleared tests. They don't have to fall under the FDA, and it is kind of a problem. Unless you're just freezing eggs, quarantining them, shipping them over here, and then they're being retested. I don't quite know how to handle international donors. I don't. Maybe someone else does.
Okay. Marlene, what's your answer to that?
We would not use like oocytes that were created in India, for example, in an anonymous situation, if they came in without any FDA testing. We would use them if all parties met, but we would not use them. But I just don't think you can view a donor as eligible if they haven't had all the appropriate testing. So you either have to say just a flat out no, or you have to find a way for people to meet, but you can't use them as an anonymous donor.
Very good. I have a question for you guys. So as we've seen in the presentation, really, and you said it's in the chat that it would be nice if you have a compliance person. This FDA compliance is really designed for large organizations, but we know that IVF centers, there are so many that are from small to medium size. And it seems a little unfair that organizations are treated in the same way, although it's the compliance, right? What's your recommendation for small organization that have, let's say, 10 to 20 employees? How can they cope up with the FDA compliance? And I'll start with you, Marlene.
We have a third party. We're not a big, at Wisconsin, we're not a big lab. So we have a third party coordinator. And so, we've sent her to, she works very tightly with the Eversana people. The FDA put out a
Guidance.
well, they do have guidance, but they did a webinar maybe at the end of last year that went through FDA requirements for donors. And so we have her watch that. I think she's watched it several times, because I got to tell you, 1271 is not an easy thing to read. And it's great nighttime reading. Puts me right to sleep. It's almost as good as organic chemistry was for me. In terms of putting me right to sleep, because it's just, for me, it's not linear. And so, there's this applies with the exception of this, and this, and then you have to go find this, and this in the law. And it's just not straightforward or easy. So we have one person who's the third party coordinator, and we've done everything we can to get her trained.
So for programs that don't really have a good comprehensive FDA manual, 15 years ago, I guess 2005, so we're talking 16 years ago, AAB had created an FDA Procedure Manual. It's a very good starting point for anyone that needs a good FDA manual. However, the specifics have to be of your SOPs have to be incorporated into the manual. The manual is very basic. It says you have to register. It says what applies. It says what to test for. And again, this manual was put out 15 years ago. I don't know if they still have it. I don't know if they've updated it, but it is a really good starting point.
Thank you so much. We have a question from the chat room. It says, male donor CMV positive, is it considered FDA eligible or ineligible?
CMV positive depends. So first of all, CMV is not considered an RCDAD by the FDA, but you must test for it. If someone is CMV positive, you need to get IgG and IgM levels. The IgG levels have to be high and the IgM levels have to be low. If your IgG is rising, then you're not quite past the point where you can be used. But if you get if you're positive, you just have to get IgG and IgM. An IgG positive, CMV positive donor is eligible, as long as they're eligible for everything else.
Fantastic. One more question from Philip. What social behavior of the donor to the FDA that FDA would consider risky to the point of being ineligible?
Oh, okay. I typed all these answers, so I don't know what happened to them.
Oh, yeah.
IV drug use, being in jail, having sex with an IV drug user, getting a tattoo with non-sterile instruments, those are considered risky. Having sex for money, risky behaviors. And your FDA medical history interview should ask those questions specifically. Have you had sex for money? Have you been in jail for more than 72 hours? Have you used IV drugs?
Very good. I have a question for you, Aman. Donors that are non-approved versus pending approval, do you quarantine reproductive tissue the same way, or are they treated differently?
Yeah, definitely treated differently. So like when I'm trying to think if we've had a case where we've had an egg retrieval, because I'm thinking of sperm, you know, we have that paperwork prior to receiving the sperm. We don't do any anonymous sperm donation here, so we would receive it from a sperm bank prior to. But an egg, I feel like we would quarantine it, like, so I can't think of a situation where we haven't had it. But should we not have the summary of records prior to the egg retrieval and us not know the eligibility, it would need to be quarantined until it's determined. So how we would handle it.
We had one early on in, you know, like back in the 90s, where the donor came in and had just not gotten her blood drawn. So on the day of retrieval, we drew her blood. And then we were still doing, even at that point, we were starting to do blastocyst transfers, usually only for leftover embryos, but we just pushed on to blastocyst and put a stat order in for all the bloods. And we had all the answers back by the time we needed to do anything like transfer or freeze. But it certainly sent everybody into a panic.
Absolutely. Carly, what are the best practices for quarantine? Could you do that exercise?
So we have quarantine. First of all, also do no sperm donors. We don't do anonymous sperm donors. We buy them from a bank. They come in. Directed donors, we use fresh with no freezing, so they're not in our system at all. So we have a quarantine tank for donors. Well, we put a lot of things in the quarantine tank. Anyone that's positive for any RC-DAD. So we have like a canister for Hep B embryos, a canister for Hep C embryos. You know, so we anything that's positive, we call it in quarantine. It's in a tank that just has like positive samples. We don't quarantine for directed sperm or anonymous sperm donors. We have quarantined some oocyte donors when we have frozen eggs. So, and again, we just have a tank in one canister in the tank that says these are in quarantine. These are eggs. This is, you know, Hep B. This is Hep C. So.
Very good. One more question on the list of infectious disease, including Zika. Now with COVID-19, is there any update on a new list from FDA, or how? What do you guys anticipate?
No, everyone should be screening everyone in their practice for COVID. You know, there's those CDC questions, but the FDA has no guidance for donors regarding COVID. And they're kind of slow. You know, they don't react really quickly. And then they'll send out some document that says, our current thinking is, and solicit responses. So my current thinking is they're not going to touch this for a while because they're busy doing other things like approving vaccines or approving tests for screening. It's not that they don't care about COVID, it's that their focus for COVID is very different. But you should be screening for COVID with some questions. Have you had a fever? Have you been diagnosed? Have you been vaccinated? Do you live with someone that's had COVID currently in the last 14 days?
Very good. Any other comments before we go to the next question from the chat? Okay, so the question probably more directed directed to you, Carly, but I think we could try to answer it from all participants if it's possible. It says, could someone comment on embryo created by sexually intimate partners, but later donates to unrelated recipients, possibly years later? How do you proceed with screening, testing, and documentation?
Okay, so we're talking about SIPs that weren't tested. The FDA strongly encourages you to screen and test the donors. You are technically not required to screen and test the donors. In this situation, the oocyte donor and the sperm donor are SIPs. You technically do not have to do it, although they use the word strongly encourage, which usually means, you know, a year down the road, it says has to. And you need to label the tissue. You need to put a biohazard sign, you need a warning, and you need to say not tested for communicable diseases. And you need to advise the recipient. So you do need to sit, and in our SOP, we actually have scripts on what you have to do to advise the recipient. You need to tell the recipient these embryos were created. They were created from sexually intimate partners. They did not have any sexually transmitted diseases at the time of embryo creation. However, they were not FDA licensed, approved, or cleared tests. So technically, according to the FDA, there's a possibility that you could get a sexually transmitted disease from these embryos, and we have some stats about what the likelihood of, even if you get a blood transfusion, you know, the likelihood of getting HIV these days is super, super low. So I think it was like 1 in 10 million. So there's, you know, so we actually have scenario scripts for completely untested embryos where we go through every single RCDAD and try to give them a little bit of information. And that way we can document that we advise the recipient. It goes in as a chart note. It's a form that we just tick off in our EMR, but it does go in as a chart note that the recipient has been advised of the communicable disease risk.
I think that's a good idea to have scripts. We don't have that, but I do think it's a great idea to do.
Right, so I did scripts for anything that had to be documented that the patient was talked to.
Yeah, we make them sign a statement that says, you know, I understand that these embryos have not been, you know, tested, or the donors for these embryos were not tested. We would ask that the patients come back and be tested, but you know, that opens its own little can of worms, because if it's been years since they created it, they could have come up with something in the meantime.
That's correct. That is correct. The FDA, we also ask them to come back to be tested. It is, the FDA strongly encourages you to have them come back and be tested, but they do not absolutely have to be tested. So if you ask someone, will you come back and let us test your blood so that we can give out these embryos? And they're like, no, man, I'm done with that. It's been 12 years, blah, blah, blah. We just have a script, patient declined retesting using FDA approved, licensed, approved, cleared test.
Yeah. One more question from the chat. It says, two same-sex female, when using their eggs as the other, using their own eggs, the other one is carrying the eggs, the embryos. They are using an eligible donor sperm from cryobank. Do they need FDA testing eligibility? Marlene?
Oh,
I know the answer. No, go ahead, Marlene. I'm not answering.
No, go right ahead, Carly. We wrestle with this all the time, and we have just decided to go ahead and test and screen the woman who's donating. We treat her like a an egg donor. So we do all the testing and screening.
Which it's okay if you overtest.
Yeah.
Like, I'm all for that. They are sexually intimate partners.
Right.
Period. They are exempted from being tested, or there are any requirements for testing. So you can take the egg from one and put it in another with no FDA screening or testing, because they are considered SIPs, which are exempt.
Yeah, we have just done that, particularly in San Francisco, where we have a lot of same-sex couples. We have just decided that we don't ever want to be in the position of having someone come back and say, "Oh, I got X, Y, or Z."
Right. Well, they should have an ID panel for sure.
Yeah.
Because like, we get ID panels on all of our patients, but we don't necessarily do FDA donor screening and testing.
Right. Exactly. We would do the same thing. Yes. But we do want all the infectious disease testing.
Mm-hmm.
Fantastic. We are launching the second poll just to get feedback from the attendees on how they feel about the webinar. We are approaching the end of this discussion on FDA regulations. I want to get your feedback or your suggestion to all the embryologists, especially the younger ones that are, embarking in this career of embryology and with all the regulations that comes with it. What is your recommendation on how to get up to date with FDA regulations? Is it really required for embryologists to do so? If so, what would be that recommendation? I'll start with you, Marlene.
Read the law. Read the law. Read it again, and then read it some more when you have forgotten what it says. And then read it again, because I still go back and read it. I have it on my computer, and I still go back and read it.
I read it all the time.
Are embryologists supposed to know the law when it comes to FDA regulations?
No, I don't think they're supposed to know the law, but if you're going to spend a lifetime in this industry, I think it's in your best interest. Because, what if you're the only person in the lab that day and your lab director isn't there and the doc comes to you and says, "What do we do with this?" You either have to say, "I don't know," or you can make an informed, or call your lab director, or you can make an informed decision. And I just think it's imperative that we all know our industry.
Very good. You mentioned earlier that you have to obviously know the law, but also don't be afraid to challenge things during inspection that feels right to you, that you know for sure that they are right, right? Carly, have you had a situation like this where you have to push back on certain things or certain?
No, I've not really ever. I mean, I have had an FDA inspector whip out the good tissue practices, and I said the same thing. They don't apply to us. You can't do that. No, and I had, up until a few months ago, 9 offices, because every office within our practice that screen or tested donors has an FEI, or an FDA registration, because they receive donor sperm, because they do IUIs in that office, and they may see donors for oocyte donors for screening and testing. So I got inspected a lot because they consider each office separate. So my biggest problem was with Zika, because we had no documentation that we had gone to a map and looked for Zika exposure. And that was when Zika had just come out and was very in the news. They wanted me to go back to every donor that had been used in that time, whatever time period it was. It was a 2-year inspection, so in the last 2 years, and go back and document from the map what the Zika. Yeah. So we did it. We sent it in. They didn't say anything else. But no, I haven't had any problems with FDA inspectors. We have, I used to hold for the entire staff FDA training. So everybody knew what to do and was comfortable with it. So.
Very good. Well, that's awesome to know these perspective, because, you know, it whole compliance is a whole science that really need you know, officers in a clinic to be able to tackle that task, and let the embryologists do embryology, right? So that would be an awesome configuration. Well, with this, we're coming close to the end of the webinar. If you guys would like to introduce your practice or your interest, and I will start with you, Amin.
Oh, thank you. My name's Amin Bayless. I'm the lab director at the Utah Fertility Center in Pleasant Grove, Utah. It's about 40 miles south of Salt Lake City. We're a 5-physician practice. One of my interests, I guess, is not FDA requirements. Luckily, I have people that have been in the field a lot longer than I have that have really held my hand and helped out a ton. And we have a third-party team who does a great job with all the regulations and helping out with inspections. So, but thanks again for having me, even though I'm the fuzzy face in a clean-shaven field right here, and I felt a little bit out of place with the FDA for sure.
Thank you so much. Thank you. If you want to introduce your practice or your interest.
Sorry, who did you?
I said Marlene.
Oh, okay, you cut out on me for a moment there. My name is Marlene Engel. I direct 5 labs right now. I'm at the currently based, my home practice is the University of Wisconsin Generations Fertility Center. I'm also an offsite for the University of Rochester in Rochester, New York, for Carolina's Fertility Institute in Winston-Salem and Greensboro. Caperton Fertility Institute in Albuquerque, and Laurel Fertility Care. I've been there since 2008. I was the on-site director for many years there in San Francisco. So my interest is keeping my head above water right now when it comes to CAP and CLIA, because, you know, it was so nice to not have any CAP inspections for 1.5 years. And man, I've got them all.
Oh yeah, they're on a mission.
Yes, they are.
Yes, yes, they are.
Very good. Carly?
So my name's Carly Chapman, and I'm currently the lab director at the Reinhardt Fertility Center. It is a new practice. It is a very small practice, and I stepped down from being the lab director at a very large practice with lots of physicians and lots of staff to manage. I spent most of my time just managing people, and at the Reinhardt Fertility Center, I'm kind of back to doing what I love. That would be compliance. Embryology. I'm actually back in the lab working, and I love it, and I am so happy that I made that decision.
Awesome. Awesome. Thank you so much. Thank you, Carly. Thank you, Marlene and Amin, for your presentation and your clarification on FDA regulation. Next week, next 2 weeks, on June 21st, we will have a webinar on current status and future of embryology and reproductive genetics. So we are going to invite some geneticists and also some people who have been involved in technology, and then just to see and to sense where the future is as far as embryology and reproductive genetics. And with this, we're coming to the end of the webinar. Thank you so much for each one of you, and I will see you in 2 weeks. Thank you so much.
Thank you.
Bye.
Bye. Thank
Bye.
you.
Everyone.